Naveta Srikanthan, and I am the project manager of the Waterloo COVID Wastewater project. So we know that COVID virus can be found in wastewater. It's shed through feces, and it travels through the system and reaches the wastewater treatment plants. So what we've been doing is collecting wastewater samples from these treatment plants before they go through any sort of treatment. We collect them over a 24-hour period, and then we transport them to our lab, where we try to extract all the viral particles and all the viral RNA that's found in that wastewater sample. And we will extract it, amplify it using the PCR machine, and try to quantify the RNA viral signal over time and try to match it with clinical data.
Hello, my name is Sarah Kowalczyk, and I'm working as a COVID research assistant in the Servos lab. When we receive the samples from each collection site, the first step is to clean the bottles. First, we wipe each bottle with 10% bleach. Second, we wipe each bottle with 70% ethanol. And finally, we UV each bottle in the biosafety cabinet for 30 minutes. The samples are aliquoted into the empty labeled tubes. This is done by inverting the bottle 5 times and then pouring 40 milliliters of influent into the tube. The samples are then kept on fresh ice until the next step. Ten microliters of internal spike is added to each tube, and then the tubes are inverted 5 times to mix. After, the samples are incubated on ice for 10 minutes. The 40 milliliter sample is now added to the tubes with pre-weighed PEG and salt. The samples are inverted 5 times to mix. The tubes are shaken until all the powder is dissolved, and then vortexed for 30 seconds at 2000 RPM. Finally, the samples are placed in a shaker at 4 degrees Celsius and shaken at 150 RPM for 2 hours. After shaking, the samples are stored at 4 degrees Celsius overnight until the next step can be started.
Hi, my name is Alice Skier, and I'm a research assistant working for the COVID-19 surveillance team at the Servos lab.
The first step is to clean all workstations. The biological safety cabinet is wiped with 70% ethanol, and the required materials are placed inside for a 30-minute UV exposure. After overnight incubation, the samples are spun in the centrifuge at 12,000 Gs for 1.5 hours with no breaks. After the spin is complete, the supernane is discarded, leaving behind only the pellet. If the supernane cannot be entirely removed, the sample is spun in the centrifuge again at 12,000 Gs for 5 minutes with no breaks. After the second spin, the remaining supernane is removed with a pipette. Sometimes a third spin is required to ensure none of the pellet is lost during supernane removal. In this case, 2 spins were sufficient. The weight of the pellet is recorded for standardization and quality assurance purposes. The samples are stored on ice until the next step can be started.
Hi, my name is Mina Hayat, and I'm a wastewater technician with the Servos group. Now that the pellets have been isolated, they must be resuspended for RNA extraction. A mixture of PM1 solution and 2-mercaptoethanol is created in a 99:1 ratio. 650 microliters of this solution is added to each sample, along with 100 microliters of triazole reagent, which helps to break down the organic materials. Aspirating the solution helps to ensure the pellet is resuspended homogeneously. The entire solution is then transferred to a 2 milliliter Power Microbiome glass-style bead beating tube. Extraction blanks and spiked extraction blanks are also included for quality assurance and comparative purposes. The samples and extraction blanks are then loaded onto a vortex which shakes the tubes at maximum speed for 10 minutes. A final spin on the mini centrifuge separates the glass beads from the sample. Samples are spun at 13,000 G for 1 minute. The final step is to load the samples into the QIAGEN QIAcube for extraction. Before samples are added, the QIAcube is prepared by adding the appropriate buffer solutions and pipette tips. 450 microliters of the supernatant is aliquoted into a rotor adapter. Solution tubes and spin columns are also added to the rotor adapter. Once all components have been added to the QIAcube, the run begins. The instrument adds solutions in series to elute the viral RNA found in the original wastewater sample. The result is a 100 microliter sample of purified RNA. The sample is stored at -20 until PCR can be performed.
Hi, my name is Kirsten, and I'm a technician in the Servos lab. The next stage of the process is PCR, or one-step reverse transcription quantitative polymerase chain reaction. qPCR allows for us to quantify the amount of our gene of interest in each sample. For the COVID wastewater surveillance project, we measure the amount of a few SARS-CoV-2 genes. We also measure the amount of another virus to use for data normalization, and internal process control, and we check for sample inhibition. The next step is to make a master mix for each target. First, we dilute the primers and probes for each gene. Then the master mix is prepared by combining water, TACpath, and the diluted primers and probes. We have several ways of assuring the quality and accuracy of each plate. No template controls are wells in which we add no sample so that we are assured there is no contamination from the external environment. No reverse transcriptase controls are prepared by taking a portion of each master mix and heating it up to kill the negative RT enzyme. This control allows us to see if there is any DNA contamination. With every PCR plate, we run a standard curve which contains a known amount of our gene target, so that we can be certain of how much gene we are measuring in every sample. To prepare the standard curve, we perform a serial dilution on a known amount of our gene target. Then, we plate the master mix, samples, standards, and controls onto a 96 well plate. We plate everything in triplicate. We seal the plate with strip caps once we are done each section of the plate. Then, the plate is inverted, vortexed, and centrifuged to mix the reactions and bring the mixtures to the bottom of each well. The PCR run is set up on the computer and the plate is loaded onto the PCR machine. In the PCR machine, RNA is first converted to DNA. Then, the reaction cycles between 95 degrees Celsius and a lower temperature, usually 55 or 60 degrees Celsius. The high temperature denatures the double-stranded DNA, and the lower temperature allows the primers to anneal to the DNA sequences and replicate the gene of interest. These steps are repeated 45 times, and each time there is a doubling of the gene of interest. As the reaction occurs, the probes fluoresce and the PCR machine measures the signal. The CT value of each sample is compared to the standard curve in order to quantify the concentration of the gene of interest in the sample. The final values are reported as copies per milliliter of the original wastewater sample.